time, believe it or not, I've been in this field for almost 25 years now. That was at a time where that actually was unclear to what extent would aging biology be conserved. And there were all the mouse people who were saying, you can only study aging in mice, and all the humans, researchers are saying, you can only study human aging in humans. And I was like, well, how do you know? And so I figured I would go out and actually test it. And so we did a set of experiments where we looked between yeast and C. elegans and tried to ask in a comprehensive way, is genetic control of aging conserved? And fortunately, it is, at least to some extent. And that's really what I focused on for the rest of my career, is which aspects of aging biology are shared as we look broadly across the animal kingdom. And that's still what I'm largely interested in. I spent about 20 years at the University of Washington as a professor, worked my way up through the academic ranks there. And then just recently, about 3 months ago, left my academic position. I am now CEO of a startup called Optispan, which is, broadly speaking, interested in creating tools that will enable the transition from reactive disease care, 20th century medicine, to proactive healthcare, 21st century medicine, where I think the biology of aging will play an important role. I'm also co-founder of a biotech company called Aura Biomedical. I'm coming to you live today from Aura headquarters, which I think, I would say is really an opportunity to move the needle in the field. I feel like the field has in some sense become narrow and very, very focused on the hallmarks of aging, and many people are not studying what we don't know. And I think Aura has an opportunity through massively high throughput longevity intervention testing and drug discovery to find out what we don't know. And so I'm super excited about those kinds of big picture questions.
Awesome. Thank you. Armand.
Oh man, tough acts to follow. Hi everyone, I'm Armand. I started my biotech career at Alnylam Pharmaceuticals, where I was their first intern. And that kind of was the thing that really got, pilled me on startups, biotech in general. After that, I did my PhD with Ben Cravat at Scripps in chemical biology. So I'm a chemical biologist. I really kind of focused on the interface of chemoproteomics and drug discovery. So I did a lot of synthetic and medicinal chemistry, and then really kind of switched into doing platform development. So making mass spec-based platforms to enable everything from, kind of fundamental biological discoveries to drug discovery. I like to say I was not working in aging, but I was maybe longevity or aging adjacent, where I think I kind of actually constant theme of my work has been platform development, enabling kind of paradigm shifts in what you can drug, and really being a really kind of effective tool to opening up new realms of therapeutics. So I did work on some aging projects. I made a drug that extended lifespan in worms, and another that was able to regenerate intestinal epithelia in aged mice. Those are both enabled by kind of chemoproteomics and covalent drug discovery. So I got to, so not like a traditional aging person, but I've definitely kind of dipped my toes in from time to time. I worked with a bunch of pharma companies during my PhD, Pfizer in a big clinical trial collaboration. I was a number of startups as well. I was on the foundational IP of two of my PI's companies, both of which have now exited, which was kind of a fun and interesting experience. And after that, I went to a flagship pioneering startup where I was like fourth or fifth employee. I built the whole proteomics drug discovery team pipeline platform to discover novel cell death signaling factors. So kind of another kind of platform build of saying, like, look, every day in your body, 500 billion cells die. And when cells die, they release their contents into the surrounding milieu, and some of those molecules will signal. And biological signaling is just a really important area of drug discovery in biology, right? And so the question was, like, across different types of cell death, like there's apoptosis and necrosis are the classics, but there's like 15 other types of cell death. There's like a new one discovered every week now. Across different forms of cell death, different disease contexts, different tissues, what molecules actually come out of dying cells and which ones signal, and can you leverage that for drug discovery? So I built a whole platform to answer that. It's relevant to a lot of diseases: aging, fibrosis, cancer, immune activation, et cetera. And while I was there, I kind of was randomly at a Y Combinator conference and I had this idea I couldn't stop thinking about, and pitched it to the president of YC at a bar. And he didn't understand it at all because I was terrible at pitching and he was not a scientist, and I probably said the word protein like 18 times. But luckily there was another YC scientist partner there who got excited. And basically, long story short, I quit my job the next day to start the company I'm working at now, which is working on next generation induced proximity medicines, which I'm really happy to talk about at any time. I could talk about it for hours, but really, long story short, it's kind of a revolution happening in small molecule drug discovery where you can make drugs that, instead of working through direct binding, work through proximity. And a really interesting lens with which to look at biology is that all biology is controlled by proximity, right? Like protein-protein interactions, even just like cells, organelles. A lot of biology happens if you put X thing near Y thing and just like force them together, and biology will happen, right? So if you can really control proximity processes, you gain a kind of toolkit to modulate biology in ways that were really not possible to do with any other modality. And so our main targets are like a cancer target, a genetic target related to Parkinson's, actually, different than Springs. But we're definitely, we think our platform is like really applicable to a lot of aging things as well, actually. So we're always happy to chat about that and the opportunity available in this space, because I think it's just like a really cool time to be in this kind of field of drug discovery.
Wonderful, thank you. I mean, to Matt's point about the people in the field of aging being a bit narrow, that's kind of a perfect example of platform that unlocks so much that it's useful to us, that people who are passionate about aging should be looking at these things and not just looking at the hallmarks of aging and reprogramming and the things that are directly connected. Okay, and last but certainly not least, we have Antoine Roux.
Hi, hi everyone. I apologize for my voice. I have a big cold, but I hope I understand everything I say. I wanted to start by thank Amos and all the Norn team for organizing this. This is really awesome. I guess I'm the only one panelist who is not a CEO. I try to do my best. So I work today at Calico. I'm a principal scientist, and I've been there from the very early days, joining with Cynthia Kenyon. So my career in the aging biology started actually a while ago, pretty much inspired by the pioneer on the field, which were like Guarantee Lab, and our very Matt Caberlin here, Cynthia Kenyon and labs like that. So I worked on yeast aging during my PhD at University of Montreal, in collaboration with several labs, including Gerardo Ferber, who's an expert in oncogene-induced senescence. So I then did some research on in yeast, on the aging. I was really enjoyed the field, really saw the potential very early, and I did my postdoc then at University of UCSF with Cynthia Kenyon, working on C. elegans, where I worked on early aspect and markers of aging in live animals. And I found really cool thing that even like juvenile, not reproductive worm could age and then rejuvenate. And I joined Calico, where today I do many things, probably too many, working on doing exploratory research in mice and human, interested in inflammation, also translational research on some potential targets. And I also try to help to keep track of what happens outside the world, in the outside world of Calico, in the biology of aging. So yeah, I'm really happy to be here and to participate. Yeah.
Wonderful, thank you, Anton. So now we're going to introduce the topic. So why are we discussing this? So my background is not in biology at all. I am a pretty new entrant to the field. I've been working in or around bio for a bit over a year now. Most of my career has been spent in and on the outskirts of tech in a few different capacities. And I am young enough and also old enough, I guess, that when I joined tech, that was a moment in which it was experiencing kind of a rapid shift in the way that work was done and how teams were structured and the kinds of roles and pathways that were available to people. So when I was joining up, user experience design it wasn't being invented, but it was being adopted. And it was still widely misunderstood and misutilized, but you started to see more user experience designers coming into companies, and the ratio of software engineers to designers, which was like this, started to shift like this. I think perhaps now there are some parallels between Wetlab and bioinformatician folks now. And you started to see all these changes. Then you saw product managers and product owners, scrum owners, all these different roles that enabled new ways of working. And you started to see teams able to start to grow larger and work on more complex problems in a more coordinated way. It allowed for more specialization of roles. And that hasn't always, or hasn't been good in every way, but it was a very interesting shift that's unlocked a lot of new possibilities. And coming into bio, I started seeing that it looks like we're at a similar time now. I think bio has lagged behind tech with respect to these things, not because the people aren't as smart or it's not as important. If anything, it's the opposite in many cases. It's more because the work is just a lot harder. And so what typically enables these shifts is the development of tools, technology, and infrastructure that kind of raises all boats and allows everyone to start playing at a higher level. And so when coming into bio, it started to look like this was kind of the moment in time that this was happening. And it's not something that happens overnight. Like I've tried to make sure we're framing this discussion in a very grounded way. I'm not saying, oh my God, change is coming tomorrow, everything's going to be different. That's not how this is going to work. But I do think that we're going to start seeing this kind of a shift. And tech and biotech, or tech bio, bi-techo, whatever the terminologies are today, I do think there's going to be a lot of change happening. And so the first question to the panelists, and whoever wants can jump on this, is, oh, damn, out of order. Sorry, folks. So so we're not doing any Q&A and anything through Zoom. Please, on your phones, go to menti.com, enter this code. This is where all of the audience engagement and all the questions will be handled. Please put nothing into the Zoom chat, only through here. That really took the wind out of my sails. Like I said, this is not me operating 100%. When you join, please give a thumbs up. We've got 45 people here, 40 panelists, so I expect to see 40 thumbs ups, please. So we can get started on the conversation while this is happening. So this is kind of what I started observing. So panelists, is this kind of true? Does it seem to you that the thrust of what I said is accurate? Is this kind of the moment that we're in bio, where tools, tech, infrastructure is developing to the point where new ways of working are possible, more interdisciplinary collaboration, et cetera, et cetera? Anyone want to volunteer?
I'll jump in and just take a stab at this. So, I mean, I think the answer is yes, but I would also say I think that's always the case, right? I mean, I think if you look 20 years ago, it was also the case that new technologies were coming online that changed the way people did biology research and biotech. And I think in the longevity field, again, I'm going to keep my answers mostly focused on the aging field, because that's what I care about, to be honest with you. It's what I'm interested in. But also, I think if you look back, I mean, and I feel very fortunate to have come into the field when I did, because it was sort of one of these moments, I feel like, where people were moving from phenomenological, observational to much more mechanistic studies using genetics and molecular biology. And that's because the tools were there to do that, and they hadn't been previously. And I think we are sort of in a similar time now. The tools, the changes are different, right? And I think, you know, the big thing that we're probably going to keep coming back to is the power of AI and how people are going to use the newly developed AI tools. And I know that's going to be a little bit, you know, it's probably going to get old talking about it, but I mean, that's the new paradigm shift, I think, that we all are aware of. And what I think is going to be the case is what we're really going to need are people who know how to use those tools and understand the biology. And I think that's what's missing, generally speaking, in biotech, but particularly in the longevity or aging field, are people or teams that have that right expertise in both the new tools that are there and a fundamental understanding of the complexity of the biology. And I think if you're missing either of those or deficient in either of those, you're going to be less effective than those teams or those people can be if you've got a good grounding in both.
So then would you characterize this as, this is kind of just the ongoing march of progress, and it's not necessarily that anything's radically different or transformative now, it's just that we have new technologies, new infrastructure that's coming along that's changing things. It's not that the pace of change is just radically transforming because of all these new things.
I do think the pace of change is accelerating, but I would say that's been happening for the last 20 years. Like if you think about the sort of hockey stick shape, right, or an exponentially increasing curve, wherever you are in that curve, it's going to feel like you're increasing exponentially because that's what's happening, right? So I'm not sure. I guess all I'm saying is I'm not sure it's different.
That's true.
Let me add to Matt's point here, because I agree completely. And I think part of it is because of the talent too. So, one thing that happened in tech is you got the best people working on the problems. And we are starting to see that. I think the longevity space has done a nice job of attracting really smart people. But besides the two things that Matt mentioned, I think the third thing that's lagging is rapid iteration. So in other types of like AI enabled tools that are being made, you can very rapidly iterate, right? And I think in science, and I'd say science broadly, but particularly in the biology, it's really hard to rapidly iterate. Even if you have an organism like C. elegans or something where you can get data really fast, it's not quite as fast as something where you can get feedback from millions of users all the time, right? So I think that's one of the challenges. I think if we can overcome that somehow as a field, that would be a major game changer as far as kind of shifting the talent to the life sciences field, because we have a lot of AI people on our team, and it's always a question of like, why work on drug discovery or life sciences when you can be working on a bunch of other things that will make you money quickly because they're lower hanging fruit, if you will.
Yeah, I mean, I think we're seeing a lot of people moving into life sciences or wanting to, precisely because we had all these the technology is being built, and initially it could really only do, well, it couldn't only do banal things, but it could like recommend songs, that would be really nice, and that's cool to work on. But now we can use this stuff for all these incredible things. Armand, it looked like I saw you unmute yourself, and that to me is the signal that someone wants to say something. So
yeah,
why don't you?
good noticing. I wanted to just also agree with what Matt said. I think there's an interesting analogy in my, so I'm a chemical biologist, which is not a field that really existed 20 or 30 years ago. And it was created specifically because there was like a bunch of biologists who were, really good at biology, but didn't maybe completely understand chemistry. And the same thing with chemists, where, you have chemists like great at making molecules, not good at understanding what a molecule needs to look like to work well in biology. And so chemical biology is different than biochemistry in that you specifically spend like a good chunk of your time doing synthetic medicinal chemistry and really understanding chemical intuition, and then using that to enable you to kind of modulate, prosecute whatever biology. And so it's truly like I really spent like 2 or 3 years of my PhD doing just pure chemistry, and then switched to doing mass spec and chemoproteomics and animal work. So I really did everything from making a drug to characterizing it to putting it to mice to doing histology and stuff like that. And I think there's a huge amount of value in something like that. I'm definitely not as good of a chemist as a true synthetic chemist, and I'm not as good of a biologist as a real biologist. But I think the intersection of those two, like another way to put this is it's very hard to be the very best in one field, but it's sometimes much easier, and you can be more unique and have more leverage if you're like top decile in two related but different fields. And I think there's that, like related to Matt's point, I think that is one thing that's missing with computational and Wetlab, where it seems to me that most people are like mostly computational, and then they kind of glom on a little Wetlab, or mostly Wetlab, and then they like learn a little kind of like coding. And there's not really this drive. There's not a lot of people who are like really like pretty good at both. And I think that is really an opportunity. I think there's just a lot of richness that you get by like having a pretty deep understanding of two kind of unrelated fields. And I think that's definitely something that is like an opportunity for people. And like, it's like, I don't even know if there's a name for it. It'd be nice if there was a name for someone who like literally spent really 50-50 between like AI computation and then Wetlab, and really kind of could then walk in kind of both fields.
I think it was a bit of a, like, I understand why initially there was like a clear line of delineation between wet lab and dry lab, but I think it is very clear that people who are working in the wet lab should probably be able to code. I mean, this is obviously up for debate, but that line of delineation, at least in the modern day, should be moving a little bit. It should be a little less clear. You should at least be able to understand these. But let's get to that in a bit. So now,
can I just jump in real quick? So I want to add a nuance here that I think is important, right? Because we think about wet lab and dry lab, and I would say those are tools, right? That's learning what the tools can do and where they can be applied. But I think there's an important thing here that I'm not sure fits cleanly into wet lab or dry lab or computational, which is how to do good science. And that to me is what's really important. How do you design experiments? how do you think critically about alternative explanations and what are the right positive controls? What's the right negative control? That's a piece that I think is underappreciated sometimes because people are so interested in learning how to do a certain protocol or how to use a certain technology that gets missed. And I just wanted to say that explicitly because I think that's really, really critical for doing high quality science.
It is important, and I'm glad you brought that up because we're talking about near future skill gaps. That's the title of this panel. But everyone, let's say in sports, everyone who's the best, they mastered the fundamentals, right? Like that's really like the foundation is just having a really strong foundation. And so that is a neat segue actually into this part of audience engagement. So I want to talk about and get the audience to participate. What are the core functions and activities of the Wetlab? And we're going to see a word map, a word cloud come up as people. You can put in, I believe, 3, but just throw things out there. We got 19 people, 18. We lost one. We have 47 people on this Zoom, and we only have we have 20. Okay, come on, people. Assay development, iteration, experimental design, cell culture. Making ideas reality. Interesting. We don't have nearly enough responses, people. Come on, throw things out there. Okay. Okay, so one thing I'm kind of curious, and I'd like the speakers perhaps to opine on this. So which of these things that we're talking about, the core functions or activities of the Wetlab, which of these are really central to the kind of work that should be done in the Wetlab, and which of these are really a product of constraints around tools and tech that have resulted in these kinds of things being assigned to Wetlab biologists? Like, are any of these things that you think are going to be taken off the plate of folks in the Wetlab? Are some of these going to be automated or farmed out? What do you think is the core? What is the core role of someone in a Wetlab across domains?
I like to say that I think the huge majority of the Wetlab that will matter for aging is happening in academia. And I think that industry is just surfing on whatever comes from academia. And I think it's a really, really important thing to keep in mind that that's really where the discoveries are made, in my opinion. And there's a good reason for that, is that it's to make discoveries, you need to make big risk. And it's hard for a startup to make big risk, and it's hard for big companies to do like long-term, research that is super exploratory. Like there's very, very few companies who do that. I mean, we're lucky to do it a little bit at Calico, but even though like we're really focused on translation. And so I think academia and keeping the Wetlab going in academia, that's what I want to say here. Yeah.
Beautiful. That's kind of a hot take, actually, because I think a lot of folks disagree and think that a lot of the more important discoveries that are being made are coming out of biotech. So I wonder if our other panelists would like to weigh in on this.
Kind of depends on your definition of importance, right? Because they're all important, but I think I agree that the kind of basic science is usually, and I don't think this is just the field of aging, but the basic science is usually done by academics, and the biotech is kind of the bridge that takes it into the translation, whether it's translating, a tool into something that is more used, or, in our case, it's translating potential therapeutics, taking it to human, right? Like it's great to do some work and understand the fundamentals, but then I would say it's equally important to take it and translate it to something that can be applied.
Yeah, I would agree with that. And I would also say I'm not sure, I'm not disagreeing with you, Antoine, but I'm not sure that academia is really all that much more high risk. I mean, having been on the in that world of fighting to get grants, I can tell you that grants are low risk, low reward. That's what gets funded. And that's just the nature of how, reviewers have been trained over the decades of not having enough funding. So I would say, from my perspective, both academia and industry are more risk averse than I would like to see them be. And I understand the reasons for that, but I think it's just a reality. But I also would agree that it's kind of a spectrum. Like, I think the discoveries all along the way are important, and it's just, where do you fall? I think you're more likely to find truly unanticipated important discoveries in academia, because not everything has to be driven by ultimately getting to a product and creating shareholder value. And maybe that's why it feels like the big discoveries are more often to come out of academia, but you need to have that sort of closer to actual real product piece that you get from biotech and ultimately pharmaceuticals as well, in order to actually create something at the end of the day that has value.
Yeah, I'm not trying to oppose those two, place where research is happening, but it's really going back to the wet lab. Like when you design an experiment in academia, you can design an experiment that is much more open, particularly in the aging field, where we still have so many things to understand. Like how does aging work in mammals? Like how really cell age in mice? Is it like chronological? Is it replicative? These kind of questions you can address much more openly in academia, where when you work in industry and you design projects, you really go after, still today, like you're going after targets, like what can I turn into a therapy? And I think it's a different way of designing an experiment at the wet lab, right? So that's what I mean actually here.
So
We'll see with Aura, because I'm telling you, if we're successful at Aura, we're going to test 1 million interventions for longevity. Now that's discovery science, and that's going to move the needle, and that's very different than what anybody is doing in academia right now.
That's true.
Yeah.
Okay, I want to move along, because there are a few other things that I want us to cover, but I want this kind of map to be something that we circle back to, or at least think about. It seems that assay development seems to be kind of the number one, because the bigger these words are, the more people wrote this. And so assay development and cell culture seem to be the two largest things there, which is interesting to me, and that experimental design is a lot smaller. And interpretation.
Amos, can I say one thing about this word map? There's one there. It was like testing something or challenging hypotheses. Whoever put that there, you get a thumbs up from me. I think that's really what Wetlab should be about, is figuring out where our models are broken and making them better. And
actually, let's not move on too soon. I'm curious, panelists, does this interest you at all? Do you think the priorities of our audience seem a bit off, or do you think they've nailed it?
I think assay development is definitely interesting, because really, essentially, I hope what people mean is making something that can answer a question, right? Making an assay that can answer a question. I'm surprised to see cell culture, because that's just like one method of getting something accomplished. There are many ways that I think you could, it's just like one method. I'm surprised to see that one be so large. I don't know what
sure,
the other
a lot of people here probably spend a lot of time doing that.
We do it too, but I mean, there are many ways to to answer
what
questions.
Do you wish was larger?
I like assay development.
Besides like what Matt said, what do you see here that seems to be underappreciated?
I like experience the unexpected, because I think for a lot of us, that's why we get into biology. It's sort of the mystery and finding out new things. I don't know that that's the purpose, but I think for a lot of us, that's what really hooks us, right? Is that curiosity. So I like that. Doesn't surprise me that I like assay development, because building new tools is important for making new discoveries, right? And that's how the technology develops. But it doesn't surprise me that there's a lot of like tool-based methodology, because that's what a lot of people, and it's not right or wrong, I just see this a lot of people are worried about learning how to do something, how to do PCR, for example, whatever, sequencing, whatever, as opposed to what can I use this tool, what question can I use this tool to answer? And so I just try to encourage the people that I mentor to think more about finding the right tool to answer the question you're interested in, as opposed to learning how to do new protocols. But I do think that's a very common sort of approach that people take.
Someone just commented, and they made a good point, which is that the question was worded too vague. And in a way that's true, but at the same time, I worded it vaguely because I wanted to see what comes to your head when someone asks this question. What kind of altitude are you at? And it seems that a lot of people are thinking kind of at like a cell culture level. They're not thinking super high level all the time. And perhaps it's good to remind ourselves of the kind of high level things, the underpinnings, the fundamentals of science. In any case, we are moving on. So we are going to be framing this discussion really around three things. So this is the first one. So it's going to be what's around, but has yet to impact biology research as much as it should. Next, we're going to be talking about what's new, and then we're going to be talking about what's next. So what's been around for a little while, but has yet to impact. So audience members, please participate, share as much as you can. What is around? What technology, disciplines, tools? Again, answer at kind of whatever altitude you want to, because we're just going to use this kind of as jumping off points for our panelists to start talking about things that are interesting. And yeah, so however you want to answer it, go for it. I expect more answers this time. Put as many thoughts as you can out there. The more we have out there, the more interesting thoughts can be sparked in our panelists' minds.
I need clarification on nature. Is that the journal or like the world around us?
Whoever wrote nature, comment that in there.
I think the journal has had an oversized impact. Let me say that, not an undersized impact. Probably more than it should.
That's a very separate conversation, but we may start that conversation on another panel. So panelists, feel free to jump in. We don't have to wait for this to go, and audience, please continue putting more things in the chat or in here as time goes on. So which of these panelists would you like to discuss?
I would say I agree with most of the things I'm looking at. I think the more interesting question might be, why haven't these things impacted research as much as they should, right? I mean,
well,
I think,
tell us.
Yeah, I don't know. I mean, I think it's well, part of it goes back to what I was alluding to before, which is that the way that at least academic research is funded, it tends to be funding incremental advances. Those are the kinds of grants that get funded, low risk, low reward advances. So I think that's part of it. I think there hasn't been enough funding for basic science to go around. And the large projects that get funded tend not to be in these sorts of truly innovative, applying, sort of new technologies in new ways. So I think that's part of it. Probably it's also getting the people with the right skills who know how to do it all in the room.
I mean, there's also the cost to it. Like single cell genomics has already a pretty big impact, but we see it every day. But actually, I think the majority of labs in the world don't even have access to it yet. So I think the big impact is just to come for things like that. We take it as granted and as like, okay, it's already a thing, but I don't think it's true for everyone.
Yeah, I mean, the sequencer is what, like $500,000 US, and that's not including VAT or import fees or shipping or anything like that. And so for a lot of people outside the US, like I've been speaking with, there's I think 1 or 2 labs in Brazil, and Brazil has lots of really actually amazing research institutions, but there are so few groups that can work on this stuff because it is just really expensive right now. That's a great point, Antoine.
Yeah, I have a couple thoughts of things that are missing that are mostly related to my biases, but I think are really important. I think one is I don't see proteomics on here, especially like activity-based proteomics. Genomics is obviously an amazing field, and data from big genomic studies is enormously useful in many ways. But it's also a few abstractions away from what's actually happening in biology, right? There's genomics, and then transcriptomics, and then proteomics, and then there's actually even beyond proteomics, it's like activity-based proteomics. So using proteomics not just to see abundance of proteins, which only correlates moderately well with transcriptomes, but actually looking at how active a protein is. And I think there's a lot of proteomics technology has gotten way better. A lot of new ways to make it easier to quantify stuff, just to detect way more things. And I think there's probably a lot of opportunities to find new targets and whatnot. Metabolomics as well. Yeah, metabolomics is still, it has not had the kind of technological shift in the last few years that proteomics has had. Still, instrumentation has gotten better, and that has helped. But I think actually the challenge of metabolomics is you just get you can't sequence nearly as well, basically, right? Because your fragmentation pattern in metabolomics is just going to be much simpler than a protein. And so yeah, I think that's actually an area where AI is potentially really useful. There's a paper about this recently of just can you use you're doing a big metabolomics experiment, you're just getting a bunch of M over Z's that you don't really know what they mean. If you could be better at identifying those and actually assigning chemical structures, that would be just incredibly valuable. So yeah, a lot of proteomic stuff, I think. So I see something about single cell proteomics. Yeah, I think single cell proteomics is also about to really take off with new just instrumentation and new ways to kind of to really crack this problem. I've really actually started to see good papers recently on like getting pretty good quantitation from a single cell, which has not really been possible in the past, or has been like really technically challenging. And then I think related again to my biases, I think a lot of, I don't really see anything about small molecule stuff here, especially a lot of the kind of revolutionary things that are happening in small molecule drug discovery, like drug discovery, DEL, those are, I think there's a lot of opportunities there, but beyond that, PROTACs, targeted protein degradation, and then what we're doing is, small molecule induced proximity medicines for things beyond degradation. And there's just a lot of targets of aging, specifically like, misfolded proteins, aggregation, et cetera, that I think could potentially be solved much better with the kind of like a PROTAC type or autophagy inducer or something like that type molecule. And like I see like a lot of aging companies, to me, it seems like it's a little overweighted in terms of like using like biologic and genetic therapeutics. And I think there's still a lot of challenges with that in terms of delivery and distribution and getting into cells, et cetera. And I think there really is like a lot of like new technologies coming online right now. And I do think it is a really special time, but especially in small molecule drug discovery, I think there is really the ability to, there are technologies out there that have the ability to really access really important targets that, in maybe a way that's much easier than doing, and cheaper too, and more systemic, et cetera, than doing with other therapeutics. So I think really, yeah, I don't really see that many papers on like PROTACs and longevity, for example. And I think, I think there should be. I'm not really sure why there isn't yet.
I'm curious whether any of the panelists, or what you guys think of. So AI is one of the bigger words on here. And of course, there are so many, these days, there are a lot of sort of AI X aging or AI X biology things happening. Does anybody think that the potential for AI to impact biology is actually overhyped in general, or maybe right now in the state that AI is?
I really don't think so. I think that, it's all about how do we treat and what do we do with our big data with, like UKBB is used massively and it's probably going to have a really great impact in the near future. And then, so, it's just a way we generate hypotheses, like Matt was saying, using this big data, we identify target. We're doing it. Calico, I think BioEdge is also a great example. And then we go back to the bench, and particularly to the animals, and then we test. And so I think it gives us a huge advantage to use machine learning, compared to our very like old school way of.
I'd like us to get a bit more specific though about what are some of the applications of the more interesting applications of AI in the wet lab that have been around for a little bit but don't get talked about. Like there are so many applications like when it comes to like labeling or imaging and all these things that are not super sexy. And so where is this stuff happening? Let's not just say AI, because AI is like not a very useful.
An example. I have a very simple example because I just used it last week. So I work with single cell sequencing data, like a lot of people, and I'm trying to identify cells in a data set. And I tried like old school method, just enrichment, markers, etc., averaging using like more fancy statistics. And I got to maybe 70%, 80% success. And then I applied machine learning, and boom, I'm like 99. And the worst case, 85, 80, 90%. So I basically made something that was not working well enough with old school math. With a machine learning, now it's like clearly working. I can use that code for my experiment, So that's a game changer, basically.
If I can give one example as well, we're using, and this kind of builds on what Armand was saying earlier about proteomics, and we talked about the other omics type of data. So multi-omics, I think you end up with like tons and tons of data. So I think AI is the only way really to tackle that kind of thing. And while I think individual headlines for AI may be overhyped, I think the field itself is very much not overhyped. And I think if anything, there's so much potential. What we're doing with it is looking at stratifying the patient population. And I think this is very true for the field of aging as well, but for any disease of aging that you look at, the patient population tends to be very heterogeneous. So not everybody kind of ages in the same way, right? So we're developing mitochondrial therapeutics. So who are the people who may benefit more from mitochondrial type therapeutics, whether it's aging or neurodegenerative disease or whatever you're talking about, versus let's say lysosomal or proteasome related targets. So I think that's an area also I'm kind of surprised not to see multi-omics, because that's a big one. And then I would also want to add human readouts. I think we see a lot in the lab, and the field of aging has done fantastically when it comes to animal models. I think we've like convinced everybody aging is modifiable, but the translation to humans is still lagging. So I think if we can have more human readouts, and this is another place again where AI can be incredibly helpful, that's an area that's definitely, I think, underhyped, if anything.
Yeah, Antoine, you were saying that you're doing some work in humans, and I'm sure you can't speak directly to what that is, and we wouldn't want you to. Well, maybe we would, but we can't ask you to speak directly to what that is. But that's actually quite fascinating to me, because there's really like almost no work on aging biology research that seems to be happening in humans outside of like geriatrics or something like that.
So what I mean by that is that you can generate hypotheses based on human data like UKBB, and then go to either like a model like, you know, animal or cell culture. It's funny that I just heard that we kind of like agree that in aging is we can tune aging in animals, and now we need to work on human. And it's funny because the way I think about it is more, okay, we know we understand pretty well aging in yeast. We understand better aging in C. elegans, where we have probably 200 or 300 genes that, or maybe 400 or 500 even, that we know can modify longevity. And we still have to do so much work in mammals. In that, my opinion is that there's still so much work to do just in mice. You know, there's a paper that just came out like I think a couple of weeks ago, where they done zero animal, only human skin aging, and they found a target. And I think this paper is really cool, and I hope it repeats. But so it shows that it's possible, but it's really hard, you know. And I think we still have a lot to learn from mice, and I think this is a bit under appreciated actually. Like I'm not convinced at all yet by the someone wrote here organoids. So organoids for aging, I think it's a great idea, but I don't think they work today. I don't, unfortunately, like we still have to understand better what happened in vivo to be able to model it in cell culture, you know. So it would be great if we could study aging in organoids, human organoids, but unfortunately we're not there yet, in my opinion.
Yeah. When you and I were talking about it, you were saying that we don't know what aging is yet, so we can't really artificially age an organoid to create the conditions in which we can actually learn as much as we might need to learn.
Yeah, I would just like to sort of add on to that. I agree with that. I think maybe where the hype gets ahead of the reality with AI. I think AI is, again, a very powerful tool, can be applied in a bunch of different ways. What you will often see are these papers that use AI to identify targets, and then that's the paper, right? They don't actually show that those targets actually are doing what we think they might be doing based on the AI identification. So they're missing the validation piece. And I think that's what Antoine is alluding to, right? Is you need to then actually go show that those targets actually behave in the way that you expect them to behave with some sort of experimental validation. And I would agree. I don't think that the in vitro or organoid-based models are sufficient validation at this point. Maybe we'll get there at some point, but I think still you need to go back to a whole animal in some way and actually show that the biology in that context is working the way that you expect it to work. The other thing I'd say about AI, and I know some people disagree with me on this, but I do also worry that some of the ways people are trying to use AI is to take what we know about aging now. So for example, what are all the genes or drugs that we know modulate the biology of aging, and use that as a training set to discover new things, right? New targets. My concern with that, that'll work to some extent. You still need to validate those targets. My concern with that is that if your training set is what you already know, you're probably not going to find anything substantially better or different than what you already know. You might find new things that work the same way as rapamycin, for example. You're not going to find anything better, right? And so my worry is that there is a bit of a blinder there, that when you're training on a limited data set that's only a fraction of the information that you really want to have, you're going to bias what you can find. So as a discovery tool, I'm not sure it's quite as powerful as a lot of people.
So Matt, I'm going to give you a nice slow pitch down the middle. So what do you think might be a good alternative to such an approach?
I think there are a bunch. Honestly, look, I've said aura a couple times. Obviously, that's fixed in my head, but I think unbiased approaches are something that the field has gotten away from. We have become very narrowly focused on the hallmarks of aging, and almost nobody is looking outside of the hallmarks of aging by and large. We should be doing that. People should be discovering new things through unbiased approaches. I feel strongly about that. I also think studying novel animals is important, right? Get outside of the 4 model systems that we use. There's a whole bunch of really interesting biology out there in other animal models that age.
What are the 4? What are the
right?
4? I'm assuming yeast, worms, mice.
Worms, flies, mice. Okay,
flies, right.
sorry, killifish people. You're not in the same class yet. Okay. No, but if you look around the field, right, those are the 4 where most of the literature exists. I certainly didn't mean to say, I like killifish, by the way. My point is that there are a bunch of other animals out there that are of interest, right? And they're understudied, and they age, at least from a longevity perspective, very differently than yeast, worms, flies, and mice. And so I think there's riches to be discovered there. It's harder to do research in those animals, but those are the kinds of things I would like to see. Just get out of your comfort zone. Don't assume that we know everything, and ask yourself, what don't we know? How much don't we know?
And I'm curious, Matt, with the model organisms and also the people over-indexing on the hallmarks as opposed to other stuff, is the reason this is happening a lot, like what's the barrier to people looking at different animals or looking at different hallmarks? Is it just a sort of, imitation situation, or is there some actual bottleneck?
Yeah, that's a great question. So, I mean, look, I can tell you what I think, which is just my opinion for what it's worth. But I mean, I think honestly, and I don't have anything against the hallmarks. I like the hallmarks, guys. Okay, I'm not bashing the hallmarks, but I think it was an unanticipated consequence of the hallmarks that when you formalized aging as these 9 or now supposedly 12 things, it gets very hard conceptually to think of any new thing that we find outside of that paradigm or that framework. And that has been built into the way that NIH grants are funded, right? If anyone here tried to submit an NIH grant to study something that didn't fit into that framework, it would not get funded. I can just tell you that, having been on NIH grant review panels. So I don't think it's anybody's fault, but I think it was then this happens as a field matures as well. It's sort of natural. In my opinion, the aging field, though, became too rigidly conceptualized at too early of a stage. And so we just need to start, we need to have people, hopefully more people like me, that'll push back on this a little bit and say there's other stuff out there to find. So I think that's partly why it's happened. It's
interesting. Our last panel had the exact opposite point of view, I think, like James Payer, Kristen Fortney, Adam Froy. I mean, not surprising to those who know them, but their perspective was, before there was not as much kind of coordination, and there was a lot of fractiousness, and that did not lead to people arguing about the right things, perhaps. I'm not saying we shouldn't keep studying the hallmarks, right? So I think this is where I don't know that I would disagree with them. All I'm saying is I think that we need more discovery science. Look, if you want to take a molecule and get it through FDA clinical trials, you almost have to be a cookie cutter at this point. You've got to do the incremental stuff. I'm not saying we shouldn't do that, but I think there's bigger things to find out there, and we need some people to be looking for that as well. Lovely. I wish we could keep talking about this all day, because I could keep talking about this all day, but we don't have enough time for this. So we're doing what is new that you believe has the potential to meaningfully change biology research. And so someone's going to put large language models in there. And if someone doesn't, then please, I'm requesting someone to put. I just read a fascinating article about utilizing large language models to study molecular biology, and it just, all this stuff seems so fast. I mean, perhaps this is not new and this is better in the next category, but all this stuff seems so exciting to me. I cannot wait to see what y'all put on there. All right, folks, back to Menti. Okay, interdisciplinarity. So interdisciplinarity is new, or perhaps our panelists can speak to this. Is interdisciplinarity sort of new within biology? Is it something that's been enabled a lot more recently? Spring, you're shaking your head.
Yeah, I mean, as somebody who was like in the farm tox department working on a neuroscience PhD, working with like biochemists and statisticians and molecular biologists, I don't think it's new. This is like
is there an extent to which it's new, in that because of new platform technology, new etc. stuff, that we can work in a more interdisciplinary way than we could before, or perhaps no?
I mean, I think in the field of aging, maybe there's a bit more interdisciplinary to it, because so many of us have come to the aging field from other fields that are maybe tangential, like, for example, the neurodegenerative space, where it's not traditional aging, but of course there's an aging factor to it. And I think that's really nice. When I went to one of the Keystone conferences, and I think everybody that got up to speak was like, well, I don't come from an aging background, but so I think that's unique to the aging field. So I will give them that. I think that's new and unique to the aging field, where you don't get it so much in the other areas.
There's also something that is probably not going to appear on that screen here, is the change of mentality of particularly the pharmaceutical industry, because the way they work today, from what I've observed myself, is that it's almost like they have a protocol on how you get from a target to a therapy, and pretty much today is like we need to understand the molecular mechanism of the disease as well as possible up to the structure, and then we'll design our drug. And when you go there as an aging biologist and you say, I'm going to study, I don't know, rejuvenation of an organ, and then make something that I'm not sure 100% how it works at this point, but I have an entry to a mechanism, and I design a drug. And so not only you're not sure about the disease yet, right, because you work on aging, but you're not sure about the full mechanism at the same point. And that pharmaceutical industry, they look at you, they just don't understand. This is completely different from their regular approach. And so I think that this day I hope is coming soon, we'll have the first drug that come from what's really tagged as aging biology. And maybe this will come, from us, from Altos, maybe from BioAge. I think the whole mentality will shift, and you will start thinking maybe they'll revise the way they go after targets and the way really they approach diseases. So yeah, I think that could make a big impact. Yeah.
Are you all seeing this kind of shift as well, Armand? This mindset shift, because you've exited and designed a few drugs, so you've been, I think, closer to this process than perhaps our other panelists.
Yeah, I think I've seen that. Yeah, I actually wanted to comment on something else, which was I love that someone put adversarial. What did they put? I lost it now. Adversarial collaboration, which I think is so important, and is really key way to answer fundamental questions.
What do you exactly do you mean by adversarial collaboration? Help me understand.
I think yeah, I mean, where I've heard this is from Scott Alexander's stuff, where it's like you have basically two people picking a topic who are both well-intentioned and on opposite sides of an issue, and then they sort of work together and directly are talking to each other, right? So it's not like one camp is saying all these things and the other camp is saying things, and it's you actually having a discussion. So key points are cruxes are being found and debated, and hypotheses are generated, and you can like get two people to agree, like if so-and-so thing is true, then I'm right and you're wrong, or vice versa. And I think that just like really doesn't happen a lot of times in biology. It's like one big group publishes a paper, and then 3 years later, another group publishes a paper, and it's like this shot across the bow, and it's just like really slow. Maybe it gets back to what Spring said about iteration, where it's like really slow to iterate on things like this because like of journal publishing speed, right? It's like you could do it on Twitter, you could do it in a journal, and there's like really nothing in between. So I would love, it would be really cool to see more of that. Actually, I love that idea. Just like maybe even like it would be cool to see a journal that does adversarial collaboration, right? That would just be amazing. So
Yeah,
love that
I
idea.
love that idea, actually. Having been involved in a couple of these things, both in journals and on Twitter, for better or worse, I actually would love that. I mean, I sort of was naive, I think, as a postdoc, and thought everybody was just trying to get the right answer, and that turns out not to be the case. But if we could have more of that, where you can actually get people who disagree to get together and just try to find the right answer, that would be fantastic. I don't have a solution for how you do that because unfortunately the incentive structure isn't always built to facilitate that. But if we could come up with a way to do it, I think that would be hugely beneficial for this field in particular.
Yeah, I joined
Honestly,
Twitter
you have
recently. Yeah, go
your
ahead.
your next task.
My next yeah, I joined Twitter recently, and the behavior of some of the academics on there I've seen is just yeah, it's lots of adversarial and not quite enough collaboration. I mean, obviously that's a generalization. Lots of folks are very friendly and lovely to each other, but yeah, it is not that way. So what about partial reprogramming? And that's the largest thing on there. How do we feel about partial reprogramming, folks? I mean, it's exciting, but perhaps overhyped. I mean, there's a lot of these companies, Belmonte putting out cryptically worded statements about, oh, we've developed this amazing thing, but behind the scenes, a lot of the scientists I talk to are pretty skeptical, not necessarily about the potential long term, but about whether there's something that's really going to be making waves in reality in the near future.
Well, they're very much dependent on the progress of the delivery today, right? So, partial reprogramming today is 4 genes, say 3 genes, and we still have a hard time to do gene therapy today, with 1 gene, and we're talking about a method that has a therapeutical window that is very small. So, there's a lot of risk into expressing these transcription factors in vivo. We know it. So I think the field has to focus on de-risking this and simplifying, like going after single transcription factors. And I think there's evidence showing that single transcription factors, gain of function, can have good impact and can have a beneficial effect for diseases. There's a paper that just came out with OCT4 alone in one of the progeria model in mice. So I think today the approach that the companies are taking is to partial reprogramming cells ex vivo and then retransplant them like immune cells, which I think is a good way to start. It's a good idea. But going after, directly in vivo, I think it will still take some technological advance. Yeah,
because I guess not all partial reprogramming is created equal, right? Like what you're talking about is perhaps a lot easier to achieve, and not the golden grail, so to speak. And so when we talk about reprogramming, I wonder, are we kind of conflating all these things into one bucket and just saying, oh, reprogramming, it's the key to everything? I mean, that's often where people's minds seem to go, at least from what I see.
There's also another thing is that we could think of loss of function of some transcription factors or inhibitors that will have the same effect of reprogramming, because that's much easier to drug than gain of function of transcription factors. Even that is still hard because it's in the nucleus. It's still hard anyway. Yeah.
So I would say one unanswered question, though. I mean, I think you're absolutely right. The technology is a limiting factor right now, right? So how do we do this in an efficient way that is going to, as you said, de-risk the potential side effects, especially if you want to get to the point where you can actually think about moving this forward into clinical trials? Obviously, that's hugely important. But I think the other fundamental question is, if we could perfect the whole transient reprogramming methodology, how good could we do? That's an unknown. And I think that's where the hype has gotten sort of far ahead of the biology, right? There are people who are throwing around words like reversing aging, where, you know, one implication is you can take an old person and turn them into a young person by reprogramming. That has not been done in any animal, right? So that's an unknown. And I think that's where the promise and the uncertainty come from. One is around, we don't have the technology now to actually do those experiments. If we could, what would be the best possible outcome we could achieve? And that's unclear, in my view, at least. I don't think we know the answer to that. Can we do better than caloric restriction or rapamycin with reprogramming? Nobody's done it yet. I think that's important for people to recognize. And in the absence of data, I think it's probably not a good idea to make it sound like that's been done already, right? So I think that's kind of where I'm at with reprogramming. We don't know yet and some of this comes back to our understanding of biology. If you believe that epigenetic changes are the overarching driver of everything in aging, then the prediction is you should be able to reverse aging. I don't know how many people actually believe that. I'm not convinced of that yet, personally.
So what else on this list do we see that panelists that you believe has the potential to meaningfully change biology research as today? What are the things that you want to highlight? And
Can I say one more thing
Wetlab.
about?
Of course.
Can I say one more thing about partial reprogramming? Yeah, just one last plug for proximity medicines. But I think the way most people have done this is, you know, genetic therapies, et cetera, right? You're trying to deliver the transcription factor, but many transcription factors, perhaps most transcription factors, are controlled by other proteins, right? Some other protein comes in and modulates it and it changes its function or translocates it to the nucleus, et cetera. And so one of our main targets at my company is a transcription factor that we've been able to essentially reprogram with a small molecule. And I think there's a huge amount of opportunity there. And this is like one aspect of what you can do. The technology we've actually built at the company is to say, you have a target you're trying to modulate in some way. And you build a phenotypic model and then scan across all potential other proteins that could modulate your target protein in a useful way. And I think we've been really interested in trying this with longevity targets. We just haven't, because a lot of reasons. Maybe don't want to get into the detail here, but it is definitely possible to reprogram transcription factors in small molecules. And I think you're going to have there's a lot of potential advantages there, right? With distribution and delivery. And I agree with what Matt said, where, you know, who knows if it's actually the right way to go, but the first step there is building the technology to test the hypothesis.
So then panelists, if you're advising someone who's an aging field or is coming up and wants to contribute to the aging field, what are some of the new kind of things that might be a bit more ancillary or adjacent than a lot of people? Because a lot of people are thinking, okay, I want to, you know, work on reprogramming, I want to work with retro, I want to work with Altos. What are some of the things that you think are new that are not really getting as much coverage? If you want to really contribute to aging research, to longevity, what are some of these side things that more people should be focusing about and thinking on? Ideally, they're on this list, but they don't have to be.
I don't know if it's new, but we haven't talked about aging clocks. I mean, I think aging clocks are something that a lot of people are interested in that already, obviously. I think there is important promise there. If we can actually develop these clocks and validate them, those are going to be, and make them better. And I guess I would put with clocks sort of personalized predictive biomarkers is maybe a better way to say it, where we can say with some confidence using diagnostics that a specific person has a specific health outcome in the future. And more importantly, test whether interventions, they could be lifestyle, they could be small molecule, modify that risk, right? If we can get to the point where we've got those personalized predictive diagnostics, that's hugely powerful going forward for the field, right? So I think that's an area where there's going to continue to be a lot of work. A lot of people are already in it, but I see a lot of promise to make advances there. And for companies, if you can validate those tests, that facilitates clinical trials, right? And again, there's a lot of misunderstanding about how FDA operates and when are these tests actually going to be useful surrogate endpoints. That's a whole nother discussion, but that's something that I think the field should be looking to. And I know people are, that maybe some of these diagnostics can be used to facilitate and accelerate geroscience clinical trials.
I mean, something that's new and that you just made me think of was, wasn't there that ALS trial or ALS drug that was able to receive approval solely on a biomarker surrogate endpoint. So is this a new development that might kind of shift the way that we're approaching these things?
Well, I don't think it's a new development. That's where I say there's a lot of misunderstanding about how FDA operates. Surrogate endpoints aren't a new development, right? You have to get to the point where you can convince the FDA or their committees that your surrogate endpoint is sufficiently accurate, right, to be very, very likely to predict health outcomes, right? Whatever your real endpoint is. So the challenge for the aging field is, how do we get to that point, right? How do we actually, if you really believe, let's just say, in the epigenetic clocks, how do you convince FDA that this is a really, really good biomarker that is in fact highly specific for telling you whether or not my intervention is actually going to improve quality or quantity of life, or whatever your endpoint is? So that's kind of a different question. How do you create that body of evidence? And that's where I think the work needs to be done.
Excellent. Now we're gonna jump to what's next that has the potential to meaningfully change biology research. These are things that might not really be around now, but that are looming, that are in development.
There is a topic in cancer that, I mean, cancer has been through like a revolution, using immune therapies. And I think that the aging field, unfortunately, we don't know enough about the very simple biology of aging to apply it the same way that is applied to tumors. But that is coming, and that's why I hope there's going to be a lot more research done, like good old biology, to really see how cells age in human, in mice, which cells are the most problematic one, which one accumulate. And then I think that's where the immune system has an importance, because it appears that cells escape clearance with age. I mean, you can talk about senescent cells, or like maybe fibroblasts that become fibrotic, etc. And using the potential of and the tweak the immune system, either using like CAR-T or in vivo, to go after those problematic cells with age. I think there's pretty big potential there. I mean, it's already starting, but I think there's going to be a lot more to come. Yeah.
So then I'm gonna insert my thing here, because we didn't get to talk about the LLM stuff that I'm curious about, and so since I'm moderating the panel, I get to talk about what I want to. So how are these? There's a lot of uncertainty about what this might actually do, and I'm very curious about how the Wetlab role. Thank you for muting. Not muting. Okay. So I'm very curious. How do we think the role of someone in the Wetlab will change in the near future? I mean, we said in the beginning of this panel that learning how to code and knowing how to program, understanding the basics of data analysis and stuff is really critical for anyone in the Wetlab. And being a hybrid scientist is really powerful. But what happens if we have these large language models that render programming expertise less relevant, and where programming expertise is less of a rate limiter? Or is that a faulty premise, in essence? Like where do we think Wetlab roles are going to be kind of moving as a result of these new tools and technologies we're having access to?
I mean, obviously none of us know the answer. I guess my intuition is that we're gonna need people who are specialized in understanding how to use these tools. So I don't think everybody in the wet lab needs to become an expert in how to apply these tools. You just need to know what they're capable of doing, and then who to go to be able to use them for whatever it is that you want to accomplish. So maybe that means there's going to be additional specialization, but I don't know that you can't be an expert in everything, right? And so I think you need to be able to understand what the new technologies can do. You don't necessarily have to know how to actually do it yourself. That's my intuition.
So maybe I can ask this question differently. So Armand, you were talking before about how it's being at the intersection of two things is really powerful. So if you're in the top decile of one thing and top decile of another thing, you could perhaps are the top 1% of those two things combined. So what what might be some of these interesting intersections that we're going to be seeing for folks in the wet lab? What are going to be some of the new pathways for people who are looking to be these interesting hybrid scientists?
Yeah, I mean, my original thing I still think is maybe my best example of just top decile of lab scientist and top decile computational person. I think that specific phenotype is very rare. I think that's, and yes, not everyone needs to be that, but I think, if you are that, it's extremely useful and valuable. And I think you just get insights into both fields that most people will lack.
And what do you think is the best way for somebody to really get into that top decile and in two or more related fields? It's not always obvious. For example, like, if you're a freshman in undergrad or a beginning PhD student, what's the best way to do that?
Yeah, I completely agree. I mean, I was very lucky that chemical biology had now been kind of formalized, and it was specifically like, you're just going to spend a few years on this and then a few years on that. And so I would just kind of try to create something like that yourself, right? Where you specifically are formalizing a program of, it's not just like in my free time, I'm going to learn coding, or my free time, I'm going to like do a few lab experiments. It's like, this is an intentional thing that I'm learning, and not sure the best way to do that, but I think it's
I'm glad you said that, actually, Armand, because this is advice that I've been giving to people who are trying to get into the field, is chart your journey and literally map it out for yourself. And right, like, here's a starting point, here's the end point, here are the intermediate points, here's what I need to get from A to B, here's what I will learn at this step. And then reach out to leaders with that kind of a map and ask for feedback on it and say, this is my plan. Because the problem when a lot of people reach out to leaders for feedback is they say, I want to pick your brain, I want to get your thoughts. It's all very general and it's not very specific. And leaders want to help. I can tell you, I reached out to Armand, Matt, Spring, Anton, and they're all obviously generous with their time and want to help. The problem is that's not very tactical. And so, if I'm receiving a message like, hey, I want to pick your brain, I may say, okay, well, is that actually going to be useful to you or to me? Like, is this a good use of time? Whereas if you chart out your journey, you say, give me feedback on this, that's really powerful. That gives something that leaders can sink their teeth into and also, respond to on their own time by email. Doesn't have to be a pick your brain kind of conversation. So I'm glad Armand brought that up, because that's a really powerful tool that I think is very heavily underutilized, is just like taking the time to sit down, structure your thoughts, and then reach out to really smart people and get their thoughts on it.
Yeah, I would agree to that. I would also say that, it's important to recognize not everybody is going to be top decile computational scientist, top decile wet lab scientist, right? You also have to figure out what's right for you. And part of this is, what do you want to do? Like, what do you enjoy? What makes you motivated and passionate? And so I would say, just be careful to try not to think that that's what you have to do, even if it's miserable, right? Because it's not right for everybody. I think that's just useful to say explicitly. And there are other ways that you can be successful and make huge contributions in the field, different from top decile and computational and wet lab.
Yeah, I completely agree with that. I would say, like, find two things that you love and become top decile on both of them is the more generalized form of the advice. So yeah.
I'm 100% backing up what Armand said, that this population that can have double skills, wet lab and data science, are rare and valuable, and we see it at Calico that I mean, it's a huge advantage if you can do that. But I think the tools. So I myself. So you were asking Amos, how do we get there if you're like a student or whatever. I'm assuming you need a lot more structure to learn the wet lab than to learn the data science. So very often we see people that are coming from the wet lab and then learn the data science afterwards. But I don't think there's like any given way. But yeah, what I want to say is that the tools that are made available today, like R, etc., are going to be more and more accessible, and thanks to ChatGPT, thanks to AI, actually. So I don't think you're going to need to be like a hardcore coder to use those tools tomorrow. So you're going to be kind of like an in-between user, and I think every single biologist, like 10 years, should have some kind of data science, and coding ability. And I think if you, 10 years from now, if you refuse to touch R or something equivalent that will be in the future, I think you'll be at a disadvantage for sure, because you'll get frustrated at one point, we use like genomics so much, and it's going to be more and more prevalent. So I will like strongly advise to young biologists to get their mind open to that. It's very important. Yeah.
Yeah, I think that's right. I would also say it depends a little bit on what you see as your career path. If you think you're going to be an academic PI versus going to industry, and where in that sort of landscape I mean, I agree completely with what you said, Antoine. I think if you're going to be an academic PI, this is from my experience as having done that. You don't have to be an expert in coding, but you need to know enough to, number one, know what can be done, and number two, at least have a feel for what the limitations are or where the pitfalls are, so that you kind of understand the landscape. And I think that's super important, but that's going to be very different as opposed to if you're going to be on the ground doing the research, needing to actually be very sophisticated in your computational skills. So that's worth just thinking about that as well. The other point I want to make, and I could be wrong, but trying to look and think about what's coming down the pipeline, and I think this is what you were alluding to, Antoine, is that I think there's going to be a supply and demand problem for people who are trained in the biology of aging and the wet lab, but I mean more understanding the biology. There are a lot more people who are very good at coding on the pure tech side than there are people who really understand the biology of aging. It's still a pretty small field, but it's growing rapidly. And so there is going to be, I think, a supply and demand in both academia and industry for people who have been trained and understand the biological piece of aging from that perspective.
Yeah.
We're right at the end of time. So I guess one question that I'm curious about is as computational research is becoming more and more central to everything biology, what are ways that wet lab folks can put themselves in a better position to collaborate with computational folks? What are some of the things that they can do to make themselves better collaborators?
Before we move on, can I counter Matt's point, which I generally agree with, but just to make it more fun. I think, as you're mapping as a young scientist, as you're mapping your future, keep in mind that you might not be right. So you may think that you want to be in academia. And Matt, I think you are an example of that. I was 100% sure I wanted to stay in academia, and then I discovered biotech and it was the best decision I made. So I wouldn't pigeonhole yourself into one role just thinking that that's definitely what you're going to do. Sorry, I still hung up on the last question.
No worries at all. So yeah, how can you be a friendlier collaborator to computational folks as an experimental biologist or wet lab biologist?
From our perspective, I think, a lot of times the computational biologists don't understand the data that well. So if you want to come from the biology side, if you have a really good, deep understanding of what the data means, kind of the big picture of what the data means, I think that really helps. From our side, we had straight up computational biologists that came in and didn't know anything about the biology of the disease that we were working on, which was Parkinson's at the time, but it could be aging just as easily. And it was very limiting. And I think the working with the just basic biologist was really helpful to kind of open that up and give you a deep understanding of the data. By the way, I'm loving all of the answers on this page as well, and loving the health biomarkers as well. Like just all of these ones, I think these are all really good. I'm excited to see what's next.
But yeah, this is something I don't want to get away from too much, because the most difficult areas. I mean, generally interdisciplinary work, it happens at the intersection, right? It happens when two folks like wet lab, computational, they hand stuff off, work hand in hand, and that's often a big challenge. Often there's kind of a gap, a chasm between them. You have a biologist who doesn't understand what it takes to analyze data. And so perhaps they designed their experiment in such a way that wasn't very friendly to the analysis later, and vice versa, where the computational folks don't understand the biology well enough. And especially now we're in a time where single cell is very popular. And yet you can use, I think there are like 4 primary methodologies you're used to do single cell analysis. And you can kind of use this different approaches and ask the same questions of the same data and get different answers. And so you need to really be able to have an understanding of the biology to be able to think critically and do it in the right way. And so how can this gap between the wet lab and the dry lab be bridged? How can people be better bridges so that fewer things fall through these gaps?
I think it all goes down to, the same old question. I mean, questions is if good collaboration is good communication, good pedagogy, and here there need to be pedagogy on both sides. Like the Wetlab needs to understand how the coder works, and how the data scientist needs to understand the biology pretty well. You need to explain very well, what you do at the bench, because that's how you together will decide where the batch effect can be like coming from. How do you like reduce it? These kind of questions, and so that's I think pretty important. And the other thing is from experience, is really to set very early on a good communication and agreement on where the collaboration is going. Authorship, the role of each person, how much time you're going to put into it, etc. Because usually data scientists are running several projects in parallel. So this very important to discuss very early on what the role of each person are, and authorship. That is a very important advice, actually.
Communication central to all kinds of relationships. That's a good point. Anything else that our esteemed panelists would like to add? This is going to be our last question, I guess, because it is now 3 minutes past the half hour.
So I'll just, looking at this list, I thought it was interesting, the aging SciComm and YouTube public outreach as potentially meaningful to biology of aging research. I agree completely. I would just say that I think that could go either direction. And so I would like to see more credible, authoritative voices and less nonsense out there representing the field.
Agreed. If I can add just one last point as well, as I know we talked a lot about the animal models, and I completely agree. There's a lot of work to be done, but I think if we can focus on human data, and that's why I was excited about the health biomarkers. I think there's a lot that we can already look at, and a lot of that data is public. So if you are a person who is interested and capable of doing computational work, just grab the data and start playing with it. Because I think that's where we're going to go. As much as even when we do the mouse studies or studies in other animals besides the 4, we're going to need to validate in humans. So I think the sooner we start with human data, the better.
Wonderful. Thank you, Spring. So the last thing, this will segue. Our next event will be this, but about computational biology. So we're going to have some great leaders in CompBio. We're going to have Jacob Kimmel from New Limit, Gerard Rutledge from Teal Omics, which is a spinoff from the Weiss Correa Lab, and some other folks. It's going to be amazing. So which computational skills or methods are you most excited about for their near future potential? We're not really going to discuss all this with the panelists. We got to let them go. They're all busy people. I thank you for giving us as much time as you did. I enjoyed the discussion a lot. Attendees, I hope you enjoyed it as well. Our next panel is 2 weeks from today, minus 90 minutes. Or 95 minutes, I guess. Where is this coming from, by the way? Where is this background?
Is it Leyland?
Leyland, I think. I think you should be able to mute them as the host.
I thought I muted every.
Yeah.
There we go.
There we go. I was hunting before. I never found it. I guess I never found it. Okay, cool. Thanks everyone for coming. If you're interested in seeing this kind of word cloud, you can stay on for a few more minutes, but we're going to be signing off. Farewell.
Hi, everyone. Thanks, Amos.
Thanks, Amos. Yeah.
Thank you, Amos.
Thanks, everyone.
Everyone.